for Neuroscience and Human Behavior at UCLA. And Michael Haro, Environmental Engineer, Senior Staff, Advanced Development Programs from Lockheed Martin.
And Kim, and thanks everyone for having me here. I'm very glad to be able to share what my passion and what our findings for our research. My name is Daniel Lee, and I'm an assistant professor in UCLA. Today I'm going to talk about basically, hopefully I can go through these two parts. The first part will be our research on Alzheimer's disease. And I think most of you, or if not everyone, knows about Alzheimer's disease, which is a very devastating disease, and it's a majority of the dementia in the elderly are Alzheimer's patients. And I would like to share first the quote by Auguste Dieter. She said, "I have lost myself, so to say." This is her response to the questions to her when she was asking multiple questions repeatedly about what she has been doing, and then she could not remember. And it was in her early 50s. She is the first Alzheimer patient being described. She sent to Dr. Alois Alzheimer in 1901 at the time, and then she, as I said, she was in her early 50s, so it is actually presenile dementia at the time, and it was very fascinating to Dr. Alzheimer, and he examined her and then kept seeing her over years, and eventually she died at 1906, and Dr. Alzheimer saw her disease progress very fast during those 5 years. After her death, her brain has been dissected. This is not her brain, but I just want to show you is how Alzheimer brain look like, and then this is a normal brain look like. So the normal brain, you can see it's very compared to normal brain, you can see a lot of grooves, which is meaning there are atrophies in the brain. And then if you have a big sections on cutting this way, and then you can see that the gray matter where the neurons are severely declined, especially in the region which help you to memorize things, the hippocampus and the entorhinal cortex area. And the pathological characteristics of Alzheimer's is what we call the plaques and the tangles. These are the deposition of a protein called beta amyloid protein, and they form these kind of like deposition because they aggregate outside of the cells and then form the plaques. And then also these it's also affects normal neuronal function and then forms these kind of tangles, which is also another protein called tau. Tau aggregates inside the neurons, and as you can see, this kind of black, or we say dark neurons, because they are not healthy at the time. So Alzheimer's disease is, majority of them are sporadic, very low percentage, less than 5%, actually is most likely 1 to 2% are familial. The way we call it familial, meaning they are purely genetic disease. If you have a mutation of certain gene, then you will get the disease. And there's only very low population, a small part of the population has that, and the gene has been identified on for this disease. And usually for early onset or familial Alzheimer's disease, the beginning or onset of the disease is around like 50 years old or earlier. And the sporadic disease, Alzheimer's usually is like 60, 70, 80, or even older. And many of these of the gene has been identified to contribute these sporadic Alzheimer's disease. So these plot I would like to share with you is the current population. You can see what we have around here. It's a population of Alzheimer's patients, and it increased dramatically over time if we don't have any cure or treatment for this disease. So currently it's like 5.2 million in the States, and the number will be doubled or if not tripled over time in 2050 if the disease is not getting control. And currently, it's about 1% of the GDP goes to the caregiving of Alzheimer's patients. And if, as I say, in 2050, that could reach $1 trillion. The single disease going to bankrupt the whole healthcare system. So that's why it's now it's a national effort. We have a lot of Alzheimer research, including the NIH and also private sectors, has a lot of fundings toward this disease because we try to find any treatment that can help not only the patients but also the caregivers and the economy. Alzheimer's disease is a multigenic disease. The reason I say it's multigenic is because most of the Alzheimer's disease patients, they are not having these three APP, PS1, and Persimmon-2 gene mutation on these three proteins. Majority of the patients does not have this mutation. They have the others, but the other gene mutation on the other genes does not give you 100% risk to get the disease. So that's multigenic. Each one contribute a little bit, and then and also your age and also your living styles and exercise will contribute to the onset of the disease. So this plot shows like the frequency. You see a very common ones are usually very low risk. As you probably heard of is called ApoE4. This is the protein for the HDL, high density lipoproteins. So you probably heard about that with the cholesterol levels in your blood. So that's ApoE4. This one is a high risk genes. If you have that, you have like 4 times more chance to get Alzheimer's disease than the people does not have it. And then today what I'm going to talk about is a gene called TREM2. This is a gene found recently, not that recently now, it was like 2013 by 2 groups. They found this also contribute a very high risk of Alzheimer's disease, even though they are very rare. And the genes I boxed out and also TREM2, they have an interesting common link to they have a common place is because they are all expressed in myeloid cells, or we say the immune cells. In the brain, there are 4 types of the cells. Neuron is what all of us know. And then also 3 other types. They are, we call them glia. They are the supporting cells in the brain. And there's microglia, astrocytes, and oligodendrocytes. So neuron, astrocytes, and oligodendrocytes, they all come from in the fetogenesis, they all come from the same resource. But microglia is actually different. Microglia is coming from outside of the brain. They are actually resident macrophages, which is part of one kind of white blood cells, but they migrate into brain when early in the fetogenesis or neurogenesis and become part of the brain. And these microglia, as I said, it's called myeloid cells. It responds to immune responses. So what the function of microglia or the job of microglia does every day is they survey the brain to look for if there's anything abnormal, if there's any pathogen invasion. And if that happens, it will respond to it and then eat them up. Or they will respond, they also will help to clean up the dead neurons. And in the embryo or neurogenesis, they will actually help to fine tune the neuronal connection. They are very, this is okay. So this is the image from, this is from culture slice, brain slice. And you can see the white ones, these are individual microglia, and these are the processes of microglia. And you can see they generally, they kind of like survey the area. They don't, the cell body is not moving, but they kind of extend their hands to touch the surface to see if there's anything going on. And later on, you will see a beam of white, or like a white dot in the middle. It's a laser. It's a laser light. So laser light burn out this area, and then we can see a microglia move into that. Let's see. So you see this green dot, and then okay, all the microglia extended their processes and also move in. So this is how they respond to the one kind of insult. Okay, so so I think I showed you how the microglia goes to to that. Then in the Alzheimer's brain, the microglia responds to the amyloid plaque, which is, as I said, these are the senile plaques, the aggregates of amyloid beta. Their job is to respond to it, confine it, and then try to clean it up. As I mentioned before, in 2013, there are 2 papers back-to-back in New England Journal of Medicine. It shows up the TREM2, there's a very rare mutation, it's about 0.9% in the population, Caucasian white. They are, if they have that specific mutation, they had odds ratio to get Alzheimer's disease to be 2 times to 4 times more than those one with a normal allele. And we know from the past, these genes only express in myeloid cells. In the brain, it's only in microglia. And if a complete loss of function, loss of this expression of this gene, it will result in a bone cyst and also white matter atrophy. But this is not Alzheimer's disease. It's just like now we know it's a partial loss of function of the TREM2 leads to the malfunction of myeloids of microglia. Okay, so now we kind of know more about what's the function of the TREM2. This is just a diagram to show how the TREM2 interact with different proteins in the cells. But I want you to look at these boxes. Initially, we know they are response to the inflammatory responses, and now we know they kind of promote phagocytosis. That means they can eat the A beta and other pathogens, and they also promote microglial survival. Similarly, in the mouse model of Alzheimer's disease, if you take out the knockout or TREM2 deficient microglia, then you can see how they respond to the plaque, which shows in green. So the red one is microglia. You see when in the brain, if there are plaque, shows in green, then microglia responds to it. So you kind of circle it up, responds to it, try to eat it up or somehow confine it. But in the TREM2 knockout mice, the microglia doesn't seem to respond to it very well. Okay, so that indicates the TREM2 may play an important role in microglia response to these kind of plaque. And we know the plaque could be the key to solve Alzheimer's disease. So the question we were asking was, okay, so can we increase TREM2 expression, and then can TREM2 expression increase will be neuroprotective against Alzheimer's disease? So what we did was to introduce TREM2 into introduce an additional copy of TREM2 genes into the mouse. By doing that, we can elevate the TREM2 expression in mice, and we'll see. Then we just interrogate how they respond to the Alzheimer's disease phenotypes. So we found it responds to the plaque very well. They kind of, you can see the green one. These are the plaques. They clear it better, so you have a reduction of the plaque. T2 is TREM2. So a reduction of the plaque when the mice overexpress or have more TREM2 expressed in the brain. And they also upregulate or increase microglial phagocytosis of the plaque. You can see that this is in the brain. You can see an increase of the phagocytosis. We're using the CD68, which is an indicator of the phagocytic activity. We see an increase of that, and also we can see that in the cell culture system. We also see the induction of the TREM2 mediated increase of phagocytosis. And then the third one is we saw a reduction of the dystrophic neurites. So you can see in the plaque in the middle, when neuron goes in, if there's a deposition of the plaque, then once the neurites goes in or in contact with it, it becomes dystrophic, meaning they are not happy, they are not healthy in that microenvironment. So you will see a lot of the dystrophic neurite shows up in red here. But with the upregulation of TREM2 expression, now we see a reduction of that. Here is the statistics. And this, not just the pathology or pathological improvement, we also see this is one kind of testing we did for memory test, of course in mouse, and they call fear conditioning, meaning they put it into a context. Basically, it's a box, and underneath there are grids. They can see around it. They remember what the box looked like and the pattern or the texture of the box. And then they were in there, but they got shot, electric shock, very mild, but it's enough to shock them. They remember. And so next time, next day, when they bring into that box, because they remember, they will start to freeze. So that's how the freezing part comes in. So we measure the freezing. That's meaning they know, and then they kind of scared about the box. But the 5XFAD is one kind of Alzheimer mouse model. They you see a reduction of freezing. That means they don't remember. They kind of like wandering around, and so they don't remember. But when we introduce more TREM2 into their brain, they remember now. So this also helps them another layer of the proof that increased TREM2 level can help. This one I want to show you is you can see normally microglia responds to a plaque. Now this in red, microglia in green. You can see they respond to a plaque like that, very dramatic change coming from here, this kind to here. But if when we introduce TREM2 to the brain, they look like these a little bit. They are not compared to the knockout, TREM2 knockouts or TREM2 deficient microglia. It is different because they still respond to it, but they tend to be less, we call it angry looking or very reactive looking. They still surrounding it, respond to it more like a normal, not engaging, plaque engaging microglia. But they tend to be able to handle the plaque much better. And we, of course, the strength of the lab is a systems biology. So we look at the whole panel of the gene expression in these mice, and then we identify these like TREM2 dependent gene cluster 1 and cluster 2. These shows up the TREM2 actually introduce a we call it reprogramming effect of the microglia. And this is just a quick diagram to summarize what I've just been telling you. Normally, the TREM2 helps the microglia to respond to the plaque and activate the microglia. But when you introduce more TREM2 into the microglia, they tend to respond it better. They kind of confine the plaque even better compared to the one with a normal amount of TREM2. And this is by different set of gene expression. Sorry. And this diagram, or this graph, we actually have a professional illustrator help us to illustrate this situation. Is normally when you are think of that there's a bushfire, and then your microglia is your firefighter. So normally the firefighter is not as experienced, and then with a small hose of the water pour into try to extinct this fire. But then if you have the TREM2 overexpression, that means you have the very well trained and then better equipped firefighter, and then you can see how big is the response of the water gets into the fire. So in light of our findings, there are actually already several company and also academia lab being looking into this. And so the first thing they did is trying to use antibody. Antibody is, they are not just like you can against the pathogens, but antibody can be used because they are very specific, recognize certain patterns. So they can be used to activate the TREM2 or help to reduce the TREM2 shedding, that meaning deletion of the, or eliminate the TREM2. So both of these can help the TREM2 signaling goes down, and then helps them to for A beta clearance. And we also have the last bit is we also did the screening by ourselves. Our approach is a bit different. We're doing that with small molecule approach. We want to use we did a screening and then try to look for the small molecules or compounds that can help to increase TREM2 levels. Okay, I guess I am a little bit out of time, so I'll skip to this.
You've got like 5-6 minutes. So
okay. Let me show you a very quick. This is another things we're working on very actively. We call it morphologic analysis. This is a thorough approach to try to use morphological analysis and a readout to for neuronal dysfunction. So this is just to show you, it's actually there are a lot of, even the neurons, we call it neurons, there are very many different types of neurons in the brain. And on top of that, we also have glial cells I just introduced you. Normally, if a neuron looks like this, during the neuronal insult or degeneration, you will start to see the neuron look like more like that. That is the function of the neuron or health status of the neuron actually can be indicated by the morphology of the neuron. So we aim to look at this to see if we can use this as a readout for a disease. But the problem is when you do the scanning try to look into individual neurons, there's almost no possible because the neuron, the density of the neuron is very high. It's just like looking into a forest, and then you cannot see which individual trees are sick. So in order to do that, we have to avoid to see everything, but we only see part of it, like sampling. And with the method we build up, now we can see the part of the neurons. It's ranging about 1% to 5% of the neurons sampled by these methods. And we can decide where, when, and what type of the cells we want to see just by genetic labeling. And this is what it looked like. Just like there are some of the neuron being labeled, and then you can see a very fine details of these like Purkinje cells. And also the microglia, they can be looked individually, and you can see the very fine processes. And we also can do a 3D reconstruction of that. Okay, and at the end, I want to show you, this is the currently our system, our platform can do a very thick section scanning and scan the whole brain. This is only part of the 500 micron sections of the brain, just to show how we can see the cells now. So you can see we fly into the brain, and then you can see individuals one. These are about 2% of the cell neurons being labeled. It's not all the cells. If you label all the cells, you cannot see this kind of very individual one. And then we can even see how they connect. So one connect to the another. Okay. All right. With that, I would like to thank all the lab members. I'm affiliated with William Yang's lab in UCLA, and there's the other the people highlighted in blue color or the people contribute to the research I've been introducing you. Okay, thank you, and I hope you learned something, and then I'm waiting for the question we can.
Yeah, no, thank you. I should admit, I did see Dr. Yang present 3 or 4 years ago, and you guys have made some like real intense progress. I'm super stunned. So I guess one of the questions is with the morphology and being able to label and see some of those neurons, but what else can we do with that? It's like what else can we do with that kind of technology?
So this is For this technology, the picture, big picture of this technology is we can each individual neuron become a single data point. So usually when we do the study, we kind of like do overall, for example, overall neuronal activity, or we do a population wise to look at how they look how they perform as a population. But now we can look into individual cells, individual neurons, and also we can look into different types of neurons at the time. That meaning the study can be more robust and thorough, but with fewer animals in hand, just to give you the same amount of the data. And the other thing is, because we're looking at the morphology into this very fine detail, we are expecting to see early changes in the morphology, which actually reflect to the cell status. No matter it's microglia or neuron or astrocytes, the morphology change is one of the indication of how they respond. So we're trying to look at that for the early signs, and so this one will help in a lot in the animal study.
Awesome. And then, okay. So another question: how do you measure the stress that was given?
Measure the what?
The stress. So when you were testing, how do you measure the stress?
So that's a very interesting question. So that's always puzzles animal researchers. Of course, you cannot. Well, I always like to say, when we study the disease, the best model is human being. But the problem is you cannot do any test on that individuals, right? So we have to use surrogates, which are the poor animals. They helps a lot. But we always talking about the conscious or the feeling. How are you going to measure them? It's always linked to the behavior we've been observing the mice or other bigger animals, how they respond to, for example, the stress as you're asking, or fear, or attention. So these are things has been studied decades, and so we kind of know what are the stress means. So for the study I showed, it was the fear and the memory. So you know that they remember because they show the fear kind of the expression. Yeah, I hope that.
Okay. No, thank you. And then a follow-up question. Sorry, multiple follow-up questions. So how can you compare the stress given to animals with kind of like normal, like, everyday human activities?
The stress?
Yeah, so like that fear stress response, like how do you?
There are actually methods to do chronic stress. One very common method of use is actually, we call it Boolean test. So you actually have we have a bigger or older male mice, and then with another one which are younger. So usually they are smaller, and the male mice they fight if they are not grow up together. They fight. So we put those two together, but not directly in contact. We have a small window, so they kind of like can contact each other in that sense. But they present over time, multiple days. So the smaller, weaker one being bullied. So that's one kind of stress. And then we can measure that using other behavior testing to look at, for example, how when they go back to the home cage or explore the new environment, how they respond to that. And when we next time introduce another new mice, not the old big bullied one, but another neighbor, how do they respond? Do they still interact, or they try to hide away? That's one way to assess the stress. But of course, there are many different methods.
And so yeah, which actually, so like it's, you know, you talked about it's like hard to like measure the actual, like, you know, some sort of like level, if you will. But, you know, but I guess the key piece really is using the same stress each on each subject, right?
Mm-hmm.
Okay.
Yeah. Yeah. There usually this kind of testing, just like we ask in the clinic, we usually have multiple methods just to try to assess one symptom. You cannot just use one to say that is right or not. You always have multiple readouts.
Okay. And so this next question is, you've shown a lot of great work from a large team. And as my colleague pointed out, because she has a PhD in microbiology, she was like, how does he have so many people? But I guess, so in order for our teachers to share with students, I guess, what are the skills that are like, you know, what are like the skills like students would need to kind of like undertake this type of a project?
So I think you probably heard a lot. Most of the people in academia will tell you passion is the core you need. You probably don't. Every time we kind of like present these kind of things, those are days, years of work, and you may only have part of the function of the figures are actually contributed by you. It's a collaborative work, and there's a lot of time just like fail and then you try again, fail, you try it again. So I think passion is one of the key. But the other thing is curiosity. I didn't get to say what when I was very intrigued by neurodegenerative disease because when I was a young kid, my uncle has a car accident and then he paralyzed because of the accident. So since then, I started to kind of like very interested into the why these kind of things happening and then why cannot be reversed. And that's kind of bring me up to so right now it's like you're always looking for the things that's very interesting to you and then you pursue it.
Awesome. And then one more question and we'll get to Michael. So like, I guess, who's on your team? Because you got a lot of people. You know, are there, you know, I know a bunch of people with PhDs, but like, do you have like you have lab technicians, data scientists you're working with? I know some people, you know, partner with other labs to do, you know, if you're trying to do a gene knockout in a mouse, like, you know, sometimes your lab doesn't actually do it, you get someone else to do that for you.
Mm-hmm. In the lab, we have a few senior scientists, which means they are already have their PhD, finished their postdoc training. We also have grad students. Currently, in the Alzheimer team, we have me and Geffen, which is another project scientist, and one grad student. And before pandemic, we actually have quite a few undergrad students. They actually contribute very well to the study. Yeah, and now we have one technician for the team.
Awesome, thank you. And if you guys are in LA, last year I had planned to do my LA event at UCLA at the Brain Research Institute, and I'm actually really hoping to do that again next spring. So maybe we can take a tour and see some of the really incredible machines that that someone else told me that you have. Apparently they're amazing.
Yeah, of course.